Hello everyone, I'm Melanie Kirch and I'm a PhD student in Phyllis T. Jones' lab at Max Planck in Tübingen in Germany. And today I would like to present our research which recently got published in Current Biology. So I'm going to talk about ancient stickleback DNA, which we managed to sequence and what it reveals about the constraints on parallel evolution. And I want to start my talk with a question, which is, how can we study the genetic basis of adaptation? And I think so far there have been two main approaches to that question. So normally when we study adaptation, we look at an organism which adapted to different environments. And then one way to learn more about this adaptation is to compare contemporary populations to each other. But while none of these contemporary populations is actually really the ancestral population, so we can just, get ideas about how adaptation works based on these present day populations. Another way to study adaptation is trying to repeat the adaptation process. So to take one ecotype and put it in a different environment and then to track the adaptation over time. But this is very challenging, like it's time intensive also, and still we don't learn so much about the original adaptation process. So how the adaptation worked for these contemporary populations that we are looking at now. So the question that is still not answered then is, what about the ancestors of the contemporary population? What do we actually know about them and how can we learn more about them? And in order to learn more about them, we established a new approach in sticklebacks, which is including ancient DNA. And so I want to start with introducing three-spine sticklebacks to you. And then I'm going to talk about this approach that we established to use ancient DNA as window back into the past. And then I'm going to answer the questions whether a contemporary population could be a good proxy for the ancestral population. And also I'm going to show you some results, some analysis which we performed with our ancient DNA, which revealed there are some constraints on parallel evolution. Yeah, so let me start with the three-spine sticklebacks. So around 10 to 12 to 20,000 years ago, at the end of the last ice age, the glaciers melted and new freshwater ponds got available. And marine sticklebacks colonized these newly available freshwater environments and adapted to the new environment. And this happened independently and repeatedly and in parallel all over the Northern Hemisphere. So from California to Alaska, in Scotland, Norway, Japan, but also here in Tübingen, in the Neckar, the river running through Tübingen, you can actually find sticklebacks. Here you can see the marine and the freshwater ecotype. And yeah, looking at these fish, would you actually have guessed that they are from the same species? Yeah, they differ tremendously, especially like obviously in morphology. So the marine ecotype is fully plated and much bigger than the freshwater ecotype, but also the freshwater ecotype normally has like a reduced pelvic spine or even lost the pelvic spine. But they don't only differ in morphology, they also differ in physiology and in behavior. And I also want to point out that the sea and the freshwater lakes, they are very different environments, not only salinity, but also temperature changes over the year, predators, the food is different, there's different parasites. So yeah, they are basically two entirely different environments. Throughout the last 20 years of research on this organism, of the genetic basis of adaptation, 242 loci were found that are consistently divergent between marine and freshwater sticklebacks, and that seem to be important for parallel adaptation. And one classical example for this is the Ida locus. So the Ida locus is controlling 70% of the variation in plate morphology. So the marine ecotype is normally completely armored, whereas the freshwater ecotype is showing reduced armor plates. And when we look at neutral markers for a global data set of sticklebacks, we can see that Atlantic and Pacific sticklebacks are grouping together, and in general, the sticklebacks that are most closely related are those that are the closest geographically. Whereas when we look at the Ida locus, we can see that freshwater and marine sticklebacks are grouping together, even if they are from entirely different parts of the world. So, and this tells us that even so they are geographically disparate populations, they have evolved this loss of lateral plates based on the same freshwater Ida haplotype. And yeah, how's this possible? Like, how can two geographically distant populations get access to the same haplotype? And what we think what happens is that freshwater adaptive alleles can leak into the marine population in contact zones and then stay in the marine population at low frequencies. So they are actually transported through the marine population by marine carriers. And then when a part of this marine population then colonizes a new freshwater habitat, they can reuse the standing genetic variation. And yeah, this is really striking because the most prevalent variant in the marine population is still the marine one. So clearly it seems to be that there's some deleterious effect, at least for some of these freshwater adaptive alleles, and still they are present in the marine population and can be reused for adaptation to a freshwater environment. And when we now get back to the question from the beginning, so how can we study the genetic base of adaptation in sticklebacks, or what approaches have been used so far? We have these two approaches to compare contemporary populations and kind of assume that the contemporary marine population is comparable to the ancestor of the freshwater population, and then also to track adaptation over time. So Mike Bell has, for example, done this in Alaska, where he put marine sticklebacks in a stickleback-free freshwater pond and then sampled the fish each year for, I think, now 30 years. And yeah, you can already, like 30 years, there's a crazy amount of time. So it's really time intensive and yeah, challenging to track this adaptation over time. And you can actually read more about this experiment in Kingman et al. On bioRxiv. And so, yeah, we still have this question open for sticklebacks, like, can we also get access to maybe the ancestors of a freshwater population? And in order to find this window to the past, we established this new approach in sticklebacks where we use ancient DNA. And for that, we looked at isolation basins. So when the glaciers melted, the land lifted up. And you can imagine this like when you get out of bed in the morning and you get off your mattress and the weight is removed from your mattress, it would also lift up again. And this is exactly what happened to the land there when the glaciers melted and the weight of the glaciers was removed. So then marine basins that were formerly connected to the sea were actually disconnected from the sea due to this land uplift. So then they turned slowly to brackish and finally freshwater isolation lakes. And the past of these lakes, you can find it in the sediments of the lakes. And yeah, also information about the organisms that used to live in this environment. So then when you take sediment cores of these isolation lakes, you can actually use it as a window to the past. So the more deeper you go in the sediment, the more you go back in time. And actually, it's really striking that, for example, for the freshwater-ish phase of the lake, we can see that the sediments are brown. For the brackish phase, they are more greenish. And for the marine basin phase, the sediments are gray. And for our study, we got 2 sediment cores from northern Norway, from Finnmark. And we got sediment cores from 2 lakes that are only 25 kilometers apart. So here Lake 1, which is called Klubvenet, and then Lake 2, Jøsavenet. And here you can see the schematic diagrams of the stratigraphy for both lakes. So, and for both lakes, we can see that the basin isolation, so the brackish phase was dated back to about 12,000 years ago. So both basins got isolated from the sea around 12,000 years ago. And the age of the stratigraphic levels was radiocarbon dated based on terrestrial plant material in the same layer. So yeah, these sediment cores were separated on 1 centimeter thick slices, and then all of these slices were sieved. And for each lake, we actually found bones were actually found for the brackish phase, and they were quickly identified to be stickleback bones. And so, yeah, we know that both of these bones are about 12,000 years old, and because they were found in the same layer, we think that they belong to the same individual. But as you can see for Lake 2, we have multiple bones, and we actually want to make sure that they are really from the same stickleback individual. So we took an X-ray scan of a freshwater modern stickleback and assigned the bones to the corresponding part in the skeleton, as you can see here with the colors. And yeah, here we can see that all these bones belong to different parts in the stickleback skeleton, and this strengthens our theory that they are actually all from the same individual. Okay, so now we had these really tiny ancient stickleback bones, but yeah, getting DNA out of these bones is actually very challenging. So first of all, because this DNA is so old, it's very fragmented. And then also you have to be super careful not to contaminate it with any modern DNA that might be present in the lab. So that's why normally when ancient DNA work is done, it's performed in this clean room environment, as you can see here in the picture, so with full body suits. And then also another challenge is that when we look at these bones, a lot of DNA that we find there is actually environmental DNA because of plants and bacteria, etc. And also the DNA that we then actually get is normally damaged and shows some damage patterns at the end of the fragments. So yeah, a lot of challenges that we had to overcome here. But Andy did a great job and got DNA out of both of these for the bones of both lakes. And just to give you an idea, even when we got sequencing data, so we got a decent amount of sequencing data for both lakes, but then when we mapped it to the stickleback genome, we actually lost 99% of the data because 99% of the data belonged to environmental DNA. So only 1% of this DNA actually was stickleback DNA. And for Lake 2, we even lost more data when we were actually removing duplicates also. So yeah, 75 megabase, that belongs about to 1/6 of the stickleback genome. So for Lake 2, we have quite a good amount of data. For Lake 1, we have very little data, and this is also why most of the analysis I'm showing later are based on the ancient genome of Lake 2. I already said that we can find DNA damage pattern in this ancient DNA, and we can actually also use it to our advantage to authenticate that this DNA is actually ancient DNA. So what happens is that cytosine gets deaminated to uracil, and this causes C to T and G to A substitutions in the sequence. And we can then measure this DNA, this substitution frequency across a fragment. So for example, for a modern sample, so on the x-axis, we can see the distance from the 5' and the 3' end. And for the modern sample, you can see that the substitutions all stay at 0 throughout the whole DNA fragment. And I actually included this little icon in the top to help you follow along whether I'm focusing on the contemporary or ancient sample in the corresponding analysis on the slide. So when we look at the same for the ancient sample, we can see for the sample from Lake 1 that C to T substitutions go up to 30% at the 5' end, and G to A substitutions also go up at the 3' end. And for Lake 2, we can see the same. And this is actually consistent with the age estimate, so what we would expect to see for such an old ancient DNA sample. And yeah, this is a way to authenticate that the DNA from the ancient samples is like endogenous and also from the ancient samples. So with that, I want to come to our questions. So now I have shown you this approach, but now what can we do with this approach? Like, we actually want to use this window back into the past now to learn whether contemporary populations are a good proxy for the ancestral populations. And for this, we looked at a global data set. So this includes Pacific and Atlantic sticklebacks and also marine and freshwater sticklebacks. Marine and freshwater is represented in red and blue colors, and Atlantic are the filled circles and Pacific the open circles. So when we performed a principal component analysis based on this global data set and including the ancient samples, you can see that PC1 is separating the Pacific and the Atlantic sticklebacks. And the ancient samples are actually clustering with the Atlantic samples, which makes sense because they are from Norway and this part also from the Atlantic Ocean. So this is what we would expect. When we looked at these 242 divergent loci, which we think is part of the parallel adaptation between marine and freshwater environment, we can see that PC1 is separating the red and the blue, so the marine and the freshwater sticklebacks from each other. And the ancient sample from Lake 2 is actually clearly clustering with the marine sticklebacks. And this makes sense because this sample is from the brackish phase. So we think that the basin just got recently isolated from the sea. So the environment is still marine-ish and makes sense that this fish is mainly carrying marine variants. Lake 1 is more intermediate, but this could also, or it's probably because we have very sparse data for the sample and we can't really get a significant result, especially because we are only looking at a subset of the genome even here. Yeah, so now we have seen that the ancient samples cluster with the Atlantic and the marine sticklebacks, but we actually want to know whether they are also similar to the local stickleback population. And for that, we got some contemporary samples from these lakes and also a nearby marine population. So we got 15 stickleback from Lake 1, from Klebanet, then 10 from Lake 2, and 5 from the sea near Lake 1. And we caught this Altafjord. These samples are called Altafjord. So when we now perform a principal component analysis with these samples, we would expect that Lake 1 and Lake 2 are separated on PC1 because they have been isolated populations. They have probably undergone some genetic drift, so they should separate on PC1. Then we expect the marine population to place somehow intermediate between the lake populations. And for the ancient samples, there are actually now two possibilities. So one is that each ancient sample clusters with the lake population. So that would mean that the ancient samples are from a time when the population already has undergone some genetic drift in the lake, and that's why it's more similar to the lake populations. And the other possibility would be that they group with the marine population and that the ancient genomes actually predate this founder effect in both lakes and are more similar to the marine present-day population. And when we actually performed the principal component analysis, we could see the latter scenario. So on PC1, you can see that Lake 1 and Lake 2 are separated and that the marine population is intermediate. And then PC2 is separating the freshwater and the marine sticklebacks. And then the ancient sample of Lake 2 and Lake 1 are also intermediate on PC1. So this suggests that they predate the founder effect in both lakes. The genome of an individual is also a mixture of alleles from different ancestries, and we used the tool NGS AdMix to get an idea of the ancestry composition for the local, but also the global stickleback populations. So here we did an analysis based on 4 different ancestries, and each column is representing the ancestry composition for one individual. We have 10 individuals for lake 1, then the ancient sample for lake 1, same for lake 2, then we have the 5 marine local fjord samples, and then also the global fish from the Atlantic and the Pacific. You can see that lake 1 and lake 2 both have one ancestry, and this makes sense because they have undergone genetic drift probably. And then we can see that the ancient samples, as well as the marine fjord samples and the Atlantic samples, show the same color, which could mean that they have a similar ancestry composition. And this gets even clearer when we did the same analysis with 3 different ancestries. So again, lake 1 and lake 2 have are clearly one color, but then the ancient and the marine samples and the Atlantic samples are a mixture of all 3 different ancestries. So they are also showing the blue and the yellow color, and this probably means that lake 1 and lake 2 carry a subset of the alleles that we can also find in the ancient and the marine samples. And this suggests further that the ancient samples and the marine fjord samples have actually a similar ancestry composition. So when I summarize now what I've shown you throughout this chapter of my talk, then while we've seen that the ancient genomes cluster with the contemporary Atlantic samples when performing a PCA, then when we only looked at the divergent loci, we could see that the ancient genomes were clustering with the marine sticklebacks. And then we've also seen that they cluster with the local marine population. Furthermore, this NGS admix plot has shown that they have probably a similar ancestry composition in the ancient and the present day marine fjord samples. So overall, we can say, yeah, at least the ancient genomes seem to be similar to the contemporary populations. And based on this limited data set, we can also say that contemporary populations seem to be a good proxy for the ancestral population. So now we've already seen that there's some data that suggests that both lakes have undergone a bottleneck, but we actually want to perform more analysis to verify this demographic history. And then I'm also going to show you some analysis on the ancient genome of Lake 2 that shows some constraints on this parallel evolution. So the genome of organisms carry a memory of what has happened to the population in the past. So the evolutionary and ecological forces that have acted upon this population. And in principle, we can use present day samples to reconstruct the demographic history that this population has undergone in the past. And this is what we did here using a pairwise sequentially Markovian coalescent analysis, also called PSMC. So we try to reconstruct the change of effective population size over time. And on the x-axis, you can see we go back in time, and on the y-axis, you can see the effective population size. And for Lake 1 and Lake 2, so the yellow and the blue line, you can see that it's decreasing towards the present. And it actually starts decreasing around 12,000 years ago. So at that time when the basin got isolated from the sea. So this is exactly what we expected to see for the freshwater populations when they undergo a bottleneck towards the present. And this is also what we have seen in other studies where they showed this PSMC analysis for freshwater stickleback populations. And well, a lower effective population size means that the fish in this lake are more closely related, and this closer relatedness, with this closer relatedness, we also expect more homozygous regions for these samples. So in order to verify this, we did another analysis. We didn't analyze the runs of homozygosity. So for 5 individuals from each location, you can see a box plot for the lengths of runs of homozygosity in their genome. And a run of homozygosity is, in this case, defined that it has to be at least 300 kb long, and we allowed for up to 4 heterozygous sites within a 300 kb window in order to account for genotyping errors. And what you can see is that Lake 1 and Lake 2 have much more longer outliers. So especially Lake 2 is showing outliers at 6 and 8 MB, which is a very long run of homozygosity. And when we summed them up, then for Lake 1, we got to 883 megabase, and for Lake 2, we got up to 227 megabase pairs, which corresponds to 18 and 60% of the stickleback genome. And to put this in perspective, so in other papers, when this number is above 10%, the term extreme inbreeding is used. And well, both values are actually above 10%. So I guess we could already also talk about extreme inbreeding here. Plus, for when you mate an individual with its parent, then for the offspring, you expect 50% of the genome to be homozygous. But for Lake 2, we even have a number higher than that. So yeah, I think this suggests that we have very strong inbreeding, especially in Lake 2. And we can find these runs of homozygosity, especially at the divergent loci in the genome. So here we look at the EDA locus again. So to remind you, this is the locus that is coding for 70% of the variants in plate morphology. And we look at a wider region here on chromosome 4, which is spanning the EDA locus. So it goes from 9 to 16 MB. And for each individual, we have one row where these bars represent the runs of homozygosity within this region. And for Lake 1, Lake 2, you can see that they span almost the entire region. And the upper part of this plot is showing the FST values in this region. So FST is a measure of population differentiation between 2 populations. The green line is showing the FST comparison between Lake 1 and Lake 2, and the other 2 lines are showing the FST comparison between Lake 1 and Lake 2 and Altafjord respectively. So what you can already see here is that the green line is a bit below, a bit lower than the other 2 lines, which makes sense because both populations have probably undergone parallel adaptation to the freshwater environment, and both carry the freshwater variants at this adaptive loci. But we can see this very strong drop, and when we zoom into this drop, this is actually like the more focal EDA region now. We can see that, especially at the focal EDA region, actually the 2 lakes seem to carry almost the exact same freshwater EDA haplotype. Okay, so we have seen now that both lakes adapted to the freshwater environment in parallel, and also that they probably have undergone some bottleneck. And now the question is, can we actually find some of these alleles that are fixed in the lake population already in the ancient genome? And for that, we looked at 813 regions that were locally divergent between Lake 2 and the marine Altafjord population. And we applied a measurement of probability of freshwater ancestry, where 0 means that the region is very similar to the marine version, and blue means it's very similar to the freshwater version. And here, each column is actually representing one region, and we rank them according for the value of the ancient genome. And as you can see here, so each line is representing one individual. So we have 5 individuals from present-day Altafjord population, 5 from present-day Lake 2 population, and then the ancient genome. As you can see here, the ancient genome is mostly carrying the marine version, but also carrying the freshwater version for some of them. But this doesn't necessarily mean that these freshwater regions have a function in freshwater adaptation. It could also mean that they have undergone some genetic drift, and that's why they are present in Lake 2 population. But if we now actually want to know whether the ancient genome was already carrying some freshwater adaptive alleles, well, for that, we looked at these 242 divergent loci again. And actually, the ancient genome was only carrying a part of these regions. So here you can see now a plot of 34 globally divergent regions. And again, each column is one region and each row is one individual, or the ancient genome. And here you can see too, the ancient genome is, well, mostly carrying the marine version at these divergent regions, but also is also carrying some freshwater adaptive regions. So we can already see that even this ancient marine stickleback is showing some standing genetic variation of freshwater adaptive alleles, even in the sparse data. So this is pretty amazing. And then we can see that, well, Lake 2 is carrying the freshwater version for some regions when the ancient genome is carrying the marine version. Well, we would kind of expect this because, well, we have sampled one fish from 12,000 years ago, but probably there was a whole population that was colonizing this lake. So some other fish were also carrying freshwater adaptive alleles, and it makes sense that some other freshwater adaptive alleles also got fixed in the lake. What is more striking, I think, is that we can see some freshwater adaptive alleles in the ancient genome, but not in the present-day Lake 2 population. So this means either they never got established in the population, or they got lost over time at some point throughout the last 12,000 years. And that means that there's maybe some constraint on parallel adaptation. And for Lake 2, we can also see it's not carrying the freshwater adaptive version for each of these divergent regions, it's actually carrying the marine version for some of them. And this could mean that, well, the individuals in Lake 2 are just poorly adapted to the freshwater environment, which will also match that we see the strong bottleneck, we see these huge homozygous regions throughout the genome. But it could also be that, that they have another set of alleles in their genome that help them adapt to the new environment. So let me sum up what I told you in this last part of my talk. So we've seen that both lake populations have undergone a bottleneck, that both lake populations also show elevated runs of homozygosity, especially at the divergent loci. Then we've seen that the ancient sample actually shows some freshwater adaptive standing genetic variation, and that some of these freshwater adaptive alleles either never got established or even got lost in the population because they're not present in the present day population. And the take-home message of this talk for you is that, well, three-spine sticklebacks are a remarkable example of parallel adaptation to a new environment. They are a powerful organism to study adaptation. Then, well, we have established this new approach to use ancient DNA extracted from stickleback bones found in sediments to give us access to a new evolutionary time point. We've seen that contemporary marine populations seem to be a good proxy for the ancestral population colonizing the lakes. And well, we've also seen that parallel evolution seems to be constrained, and not all freshwater adaptive alleles are present in a local population and could have gotten lost over time. Our upcoming plans with this project are to sample more sediment cores, to drill multiple cores from both study lakes in order to hopefully get more time points and also more individuals for each time point. So now we only got one individual for one time point, and it would be nice to get more of an idea how the whole population at one time point could look like. And yeah, you might have realized that, well, the most challenging part of this project was actually to get enough material out of this ancient DNA. So for the next samples, we are planning to use a new single strand library build method in order to improve the genomic coverage, and also, to just be able to perform better analysis based on this ancient genome. And then we also want to further study the impact of demography on fitness based on some modern samples. And with this, I want to end my talk and thank you for tuning in and listening. And of course, thank you to everybody that contributed to this project, especially Andy Fudge, who had the idea of studying ancient stickleback bones and who brought us all together to work on this project. But also, thanks to Phyllis T. Jones, to Tom Gilbert and Anders Romonset, who all contributed in various ways to this project. And last but not least, of course, I want to thank our funding sources. Without them, this project would have been impossible. And thank you. And I'm happy to take any questions.
Thanks, Melanie. That was really great. And unfortunately, we don't have a live audience, so it's just me asking the questions. And what we did last time is I set this limit of 3 questions, which I think I then immediately broke. So when I was speaking to Patricia, I went on for about 4 or 5 questions, but I actually wrote down 3 as we were going through. So I'm reading from my phone here, if you see me looking down. So given that the ancient samples clustered with the present day marine samples, what do you think we learned from the ancient stickleback genomes that you couldn't have learned from just simply comparing the present day marine and freshwater samples?
So I think it's just that the ancient sample is actually from the same lake as the contemporary population. So when we would have compared it just to the marine contemporary population, we wouldn't have realized that some of the freshwater adaptive alleles, for example, didn't get established or got lost over time, because we wouldn't have this time point back for the same lake, and to actually know that, well, this allele was present in this lake at this location, and then got lost over time at some point. I think this, for example, something that we wouldn't have realized when we just would have compared it to another marine contemporary population.
Yeah, I think that's a really nice finding, and it's sort of quite a strong contrast to some of the, for example, the experimental studies you mentioned by Mike Bell, and there's also been work done by Rowan Barrett and Dol Schluter, where we've seen an adaptation happen quite fast in just a few generations. And I guess it shows that, like there's all these different variables that could maybe act as a constraint on that, which you did a fantastic job of highlighting. So I guess, do you think this is going to sort of be a more of a, what's the word, like a less of a novelty and more of a, what's the word, something like a new weapon in the arsenal, like that evolutionary biologists will start using ancient DNA for studies like this? So do you think we can expect to see more ancient DNA stickleback studies, and what do you think might come out of doing that?
Yeah, I hope so. I mean, because while there are a lot of studies on sticklebacks, and I think it's just a new approach to study this model system. And I hope that we can, that there will be more studies, and that we can actually also get more of an idea when we get more samples from the past. So whether an ancestral population is actually similar to the marine population, like to get more verification for that, but also to get multiple time points and actually to see over time, for example, when we have a sample in the freshwater phase, like is this fish in the past then already adapted as good as the present day fish, or to get more of an idea of the speed of adaptation when we have like real time samples. So I think it's a really powerful approach.
Yeah, I think that's the key thing with all these ancient DNA studies. You're getting insights into the chronology and the tempo that you just couldn't get from present day samples. And who knows, maybe it'll give us more insights into the selection coefficients at different loci and so on. So it's really cool. I hope we can expand it more. So I guess the last question, just a little bit about yourself. Like you have like maybe just about a year or so left now on your PhD, and just wondered, are you allowed to talk about the other things you're working on, or is it kind of top secret? Or?
No, I can talk a bit about that. So I'm actually studying standing genetic variation in contemporary populations, and I'm trying to quantify the standing genetic variation. So for that, I have sequenced 10,000 marine sticklebacks at these 242 divergent loci, yeah, which is a huge data set. And we're trying to answer questions like at what frequency we can find freshwater adaptive alleles in this population, and whether all of these fish carry a certain amount of freshwater adaptive alleles, or whether there are just a few carrying a lot of freshwater adaptive alleles, whether there's some linkage disequilibrium, or how big the haplotypes are that they are carrying. So yeah, trying to learn more about this, yeah, standing genetic variation, also how it constrains or helps rapid adaptation.
That's amazing. Wow, gosh, it's such an incredible large-scale study. So I guess that people that want to keep track of what you're working on, probably the best thing is if they follow you on Twitter. And what we'll do is we'll put a little link to your Twitter name in the details of this video, and also the link to the lab website as well, so they can look at all the other work that's going on in Felicity's lab. And there's lots of really cool stickleback related studies going on. So again, it just remains for me to say thanks very much, Melanie. It's been an
Thank you.
absolute pleasure.
Thank you for having me.
Work with you. And this, yeah, thanks for giving us such a great presentation today. And that's it. So I will stop recording.